Our bellwork question for today is, what is asexual reproduction? Based on the name there, asexual, we know that there is only one parent involved. A meaning one parent. If there's only one parent involved, you're going to have the exact same genetic information as that parent. There is no other parent to be giving genetic information to the offspring. Our fact for today is that embryonic stem cells can produce all 216 kinds of human cells. So when there is first fertilization of the egg by the sperm cell, the cells that develop are going to be the embryonic stem cells, which will be able to turn into all of the different kinds of cells that they need to be in order to form a human baby. By the end of this lesson today, we need to be able to list the stages of mitosis in order and explain what happens in each of those stages. So mitosis is the type of cell division that lets asexual reproduction happen. You're going to get an exact copy of the parent cell. Now, this is really important for, obviously, for asexual reproduction, but it's also important for the cells in the body. If you have a skin cell and it needs to reproduce, you want another skin cell forming from that process. So we do want an exact copy to be made. If you're making a muscle cell to replace an existing muscle cell that's damaged or worn out or whatever, you're going to, again, need a cell that's exactly the same. So you want this type of cell division that will allow for that exact copy to be made. So the process that the cells go through is called the cell cycle. They grow and then they divide. And there's 3 stages to this process. We have interphase, then we have mitosis, where the cell is dividing, and then cytokinesis, where they separate. As we go through these different stages, I'm going to be giving you some ways to remember these, some little tricks to help you remember. So the first stage is interphase. This is where the cell spends most of its time. So it happens from right after cytokinesis, which is where the cell is split into 2 separate cells. And during this time, the cell is getting ready to divide again. It's growing, it's maturing in size, things are doubling. It's making copies of the DNA that it holds in its nucleus, and it's getting ready to divide again. So the first stage in the cell cycle is interphase. Then the cell gets ready for mitosis. The reason cells have to go through mitosis is to allow the organism to grow and to replace the dead or worn out cells. So when you were born, you had a set number of cells. Those cells didn't just get bigger to allow you to grow, you actually increased the number of cells that you have in your body. Also, I like to say we've got a similar characteristic as the Wolverine from X-Men. We're able to heal our bodies, obviously not as fast, but if you break a bone, it will heal. If you cut yourself, it will heal, usually. So we need the mitosis for the growth of these organisms and to replace those dead or worn out cells. And mitosis, that process of cell division, gives you 2 daughter cells from the one parent cell that are exact replicas and copies of the parent. So the cell is going to get organized and ready to divide. Mitosis is where the nucleus is dividing into 2 new nuclei before the cells completely break apart. During interphase, remember, it's copying everything. So it has a double amount of everything in the cell. So that way when it divides through mitosis, and eventually cytokinesis at the very end, each cell will have what it needs. So mitosis is where the nucleus is dividing, putting 1 copy of DNA into each of those daughter cells. And within mitosis, we have 4 steps. There's prophase, metaphase, anaphase, and telophase. I've heard it pronounced telophase as well. So telophase, we have 2 nuclei in 1 cell. Anaphase, things are moving apart. Metaphase are lining up in the middle. And then prophase, we're starting to see the chromosomes starting to form. So prophase, this is where we start to get rid of the, we start to get the chromosomes forming. So the DNA, let me go back a slide here, it's just kind of a jumbled mess. And it realizes, oh, we got to get ready to make sure that each part of the DNA gets into each cell. We need a copy in each. So instead of having this big jumbled mess of DNA, it starts to coil up on itself and it forms these X-shaped chromosomes in order to make sure that when it divides, the information, that genetic information, gets where it needs to go. The DNA starts to form those chromosomes, okay? We call that X shape a chromosome. So when you have prophase happening, you're starting to see X's are forming. Those are the chromosomes. The chromosome is a double rod of DNA, carries genetic information. Each piece of it is called a chromatid. So you've got one side's a chromatid, and then they call it the sister chromatid, and they're connected by a centromere. You're going to need to know this for biology next year. So each side is an exact copy. Then the process needs to have these chromosomes line up through the center of the cell so that we can pull half of that X that has two exact copies, one on either side of the X. We need to pull those apart so each new cell has a copy of that genetic information. So in metaphase, they line up in the middle. Meta, middle, they're in the middle. We have these little lines that you can see here in this picture. Those are called spindle fibers. They attach at the centromeres. That's what's going to pull these chromosomes apart. So prophase, usually if you make like a prototype, it comes before you actually build the design that you have. So proto meaning before. So prophase, we're making those X's. Metaphase, they line up in the middle, meta, middle. Then we have anaphase. This is where those chromosomes get pulled apart. So one copy, one side of that chromosome that has one copy of the genetic information gets pulled to one side. The other side of that chromosome that has the exact DNA gets pulled to the other side. And then those cells that will result from this will have the information that they need. So anaphase, they pull apart. Meta, middle, ana, apart. Anaphase, apart. And you can see in the cell, you kind of see this like V shape to these because the chromosomes are being pulled in the middle. And then we have telophase. This is where we have two nuclei within the same cell. So telophase 2, you have two nuclei in the cell at the same time. So there's a nuclear membrane that forms around each of those new sets of chromosomes. Those chromosomes also will start to unravel and stretch apart back into what we had seen at the beginning during interphase. And then finally, we have cytokinesis. This is where the cell completely pulls apart into those two new cells with their own nucleus and everything else they need to survive as an independent cell. So the animal cell's cytokinesis kind of looks like taking a balloon, a blown up balloon, and just pinching the middle of it till the two sides of the balloon touch. You get a blob on one side, you get a blob on the other side. It just pinches right down in between them and the two cells separate. Now each of these daughter cells should have an exact copy of the DNA and half the organelle from the original cell. The plants, we know, have a cell wall, which means there's an extra step for cytokinesis. They have a cell plate that forms through the middle of the cell, and then you get the cell membrane to form on either side of it, and you get the new cell wall to form. So it's not going to be the pinching like we have in the animal cell. We have a cell plate that has to form first. So one way to remember the cell cycle order is to look at the first letter of each of the stages. We have interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. So I, P, M, A, T, C. One of my students a couple of years ago came up with this saying to help us remember the order, and it's I play music at the concert. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Let's see what this looks like as the cell goes through this cell division process and through the cell cycle. So we've got the stages of mitosis. We go into the cell. You can see the organelle here. There's some Golgi bodies. There's the nucleus down here. We're going to go inside of it. The endoplasmic reticulum is surrounding it. So the DNA starts to clump together and form those chromosomes in prophase. Now they're lining up in metaphase, they're meta-middle. Those spindle fibers are attaching at the centromeres and they're going to pull them apart, which would be anaphase. So you can see them pulling apart, look like little V's almost. Now we have nuclear membranes forming around them, so that's telophase. And then the cell is going to pinch down through the center. You can see it's kind of squeezing together, and now we've got cytokinesis where the two new cells have formed. And again, they're going to be an exact copy of the other cell. They have the parent cell, and then that pulls apart into two new cells that are identical copies of what that original parent cell had within it.